Thank you, Rongroi. Hi, everyone. This is HyFlux Studio, a desktop workspace that turns hydrogen permeation measurements into fitted, traceable, and comparison ready results. In other words, it handles the part between the mass spectrometer, produced a spreadsheet, and please explain this plot at group meeting. Everything runs locally, so your data does not take an unexpected holiday in the cloud. Behind the interface, HyFlux Studio is built from independent modules for validation, processing, fitting, and reporting. Everything is connected through SQLite with experimental data, analysis settings, and results stored in a single .db file. So backing up the project or moving it to another computer is basically a one-file job. The key algorithm starts by selecting a step-specific baseline and converting hydrogen counts into atomic hydrogen flux. Smoothing is scaled to the observed transition time. Rapid changes keep short support, while stable regions can use wider Savitsky-Golay windows. Adaptive sampling then keeps extra points where the signal changes fastest without letting the quiet regions disappear. For fitting, HyFlux uses analytical rise and decay solutions of fixed second law. Each rise must establish its own stable steady state flux, JSS. With that plateau fixed, DApp is fitted across the main part of the transient. A global scan in log D finds the right basin before refinement. A decay reuses the JSS from its matching rise. The software also checks sensitivity and R squared because one cheerful looking curve is not yet quality control. Now, let us use it. The home screen has three main routes. Add data starts a new analysis. Browse data finds and manages existing tests. Overview compares steps across the database. The small book icon opens a parameter dictionary, which is useful when a symbol has started behaving like a secret password. Click Add Data, then Lab Gaseous. The other types of function, for example, the electrochemical experiment, are future modules. It is also hoped that functions for the collection and analysis of literature data will also be integrated in the future. Today, gaseous lab data is the working route. Enter the sample and test details. In the step schedule, paste cells directly from Excel. Use next start as end completes consecutive time windows. Fill T copies the selected temperature, and fill R/DPH2 fills paired rise and decay pressures. Save the form as a reusable template, so repeated experiments involve less repeated typing. Processing settings can stay on default for normal use. Custom mode exposes the baseline window, smoothing level, sampling mode, point budget, and ignored early fit time. Every combination becomes an immutable analysis profile. So trying a new setting creates a traceable result instead of quietly overwriting the old one. Continue to raw data. Choose the original mass spectrometer Excel export or use manual import and paste a table. Extra gas channels can be mapped too. HyFlux checks numeric values, time order, required channels, step coverage, and duplicate files before processing. It is friendlier to reject a bad row here than to admire a beautifully fitted mistake later. During processing, the progress page shows the current stage and log. Stop processing requests a safe cooperative stop. Completed steps are kept while the current uncommitted step is rolled back. The result opens on summary. The left plots compare this test and show the sample history. The right-hand tiles give a quick check of every transient. Choose a step to inspect DApp, JSS, R squared, fugacity, permeability, diagnostics, and the fitted curve. Click the diffusivity or permeability heading to cycle through calculations. Plot points changes display density only. It never refits the data. Each export icon saves exactly what that plot shows. Use reprocess to create another analysis profile without importing the raw file again. Select Use an Overview when that profile should represent the test in comparisons. Back in Browse Data, search, filter, sort, rename, open, or safely delete tests. Finally, Overview lets you select multiple steps and drive 4 independent plots. Each plot can show fitted parameters or experimental and fitted curves with its own axis, point count, and export button. That is HyFlux Studio. Fewer spreadsheet rituals, more reproducible science, and hopefully fewer late-night arguments with Excel. And if you want to try it, there is no Python, no IDE, and no environment setup required. Just download the Windows package, unzip the complete folder, and run h2permeation.exe. Give it a try on your next permeation test and let me know which workflow we should automate next.